This podcast is part of a special series highlighting work to be discussed at the 2024 Cancer Neuroscience Symposium, which is hosted by MD Anderson Cancer Center and Wiley on February 28th through March 1st, 2024, with options to attend virtually or in person. Learn more and register at mdanderson.org/Cancer Neurosymposium.
Hey everyone, welcome to Beyond Cancer today. I'm your host, along with Moran and Brandy, and today we're interviewing Dr. Demir.
Yeah, well, today we have the honors and the special opportunity to have Dr. Ekin Demir from the Technical University of Munich. Dr. Demir got his medical training at the Heidelberg University in Germany. And he completed his training in 2011, and currently he's serving as surgeon scientist at the Technical University of Munich as a professor of surgery. He has a very unique area of research, which I'd love to hear about from you, Ekin. You're tackling cancer neuroscience from a very unique angle of the fourth root of metastasis, which is perineural invasion. Can you please tell us a little bit about your journey and your research?
Yeah, thank you, Moran, for the very kind introduction and for the background. And it's my great pleasure to be here in this podcast organized by you. And right at the beginning, I want to say that I was also one of the participants of the first Cancer Neuroscience Symposium that you organized, and already looking forward to the second one. And I think the main reason is that this field is growing so rapidly in the last 5 to 6 years, I would say, because I entered the field a longer time ago, as you suggested. So when I was a medical student back in 2004, my mentor, Professor Gurab actually, who was my advisor for the doctoral thesis, showed to me back then some images of rats that had pancreatitis, acute pancreatitis. And he said that he doesn't look at the pancreas, but actually looked at the sensory neurons of the pancreas in these rats. And so when I said about why exactly you are looking at this, he said, look at these changes that we observe. He showed him images of dorsal root ganglia that were stained for some neuronal activity markers like CFOS and phosphoric. And showed to me that when the animals have acute pancreatitis, all these neurons get activated and all these markers are upregulated. When I first saw these images, which were back then really high quality, quite impressive immunofluorescence images, I thought that it is really an intriguing idea to look at two systems simultaneously in a research field. Because as a medical student, you can imagine we learn about the digestive system in a period, and then we learn about the nervous system. But I think back then it was, at least in my training, it was such that we did not really learn about the intersections of these different systems and how these different systems interact. And when I saw these images, I thought that this is a research field where they look at both systems simultaneously and how they intersect, how they interact. And this was the moment where I got fascinated, actually, for the interaction of the nervous system with the digestive tract. And over time, we looked not only at, let's say, a murine pancreatitis, but we started looking also at murine cancer, especially pancreatic cancer. And he then showed to me images of perineural invasion, as Moran suggested. Perineural invasion is clinically a big problem because every pancreatic cancer patient has perineural invasion, and the ones with perineural invasion are those who have the worst prognosis among all pancreatic cancer patients. And whenever I, even today, see a cross-section of a pancreatic cancer, I'm still, after nearly 20 years, still very much impressed by the extent of the infiltration of these nerves by cancer cells. So these images and the way he attracted me to this field was the beginning of my journey, coming to your question. And since then, I'm still working in this field. Of course, nerve alterations are very much related to the emergence of pain in these patients. And pain is definitely by far the most unpleasant symptom in these diseases. And so pain mechanisms were one focus. But having practiced medicine and surgery, let's say in the past, I would say 14 years, I've repeatedly seen that one of the biggest clinical challenges, especially in cancer patients, we have is that we cannot control this nerve invasion. Because whenever we operate on these patients and take out these cancers, we can never take out all the nerves that actually go into these pancreas. This is technically actually not possible. And as long as we do not control nerve invasion, I always get the feeling that all the surgical efforts, all the perfect operations that we do will not eventually lead to a cure. And this is something that still motivates me today from a translational science perspective to go deeper in this field and to delve deeper into this problem. So I hope that, that's at least in part answering your question. And my current research is, I would say, mostly therefore focused, as you also suggested, on perineural invasion, but also, of course, some other aspects of nerve-cancer interaction. I can go into more detail if you wish.
No, this is great. We've been interviewing in this podcast many of the other players in the field, which you're familiar with, and many of them are talking about the direct regulation of cancer by nerves. And it's really refreshing to hear both from you, by the way, and Rich Wong about how nerves are serving as highway for dissemination. We all intuitively familiar with hematogenous spread and lymphatic spread and local invasion, but it's not really trivial to think about nerves as highways for dissemination.
Yes.
And I think one of the reasons is that the nerves just doesn't have a lumen. It's easier for us to imagine that those cancer cells disseminate through vessels or like pipes, but nerves they don't have a lumen. So how would you consider that even feasible to travel along a route that doesn't have any space or capacity to accommodate that?
Yeah, that's a very very unique and very intriguing question. In the past, people thought that there are like some small lymphatic openings that penetrate through the barrier of a nerve, and that the cancer cells go through those really tiny openings. But electron microscopy studies showed that these openings probably do not even exist. And our research also from the past 5 to 6 years, which is yet to be published actually, has shown that nerves, especially the perineural sheaths, but also the Schwann cells along the axons, present to cancer cells a lot of attachment sites. And these attachment sites are quite comparable to, let's say, some molecules that are normally present in the extracellular matrix, in the stromal of a tumor. And we see a really high concentration of these attachment sites in the perineurium and on the Schwann cells. So we believe that it is not, the necessity for a lumen for it that enables the cancer cells to spread, but probably the sole existence of a high concentration of attachment sites enables them to walk around or along this nerves for cancer cells, for them to spread out of the organ. So I think even though the nerves lack a lumen, we believe that it's those attachment sites that make it all possible.
Yeah, and it's really great to see how those paradigms changed, right?
Yes.
Like you said, people thought about lymphatic spread because we didn't have any other explanation, or the locus minoris, or the point of least resistance. But all those notions has been proved to be wrong. Thanks to work led by people like you and Rich Wong in Memorial Sloan Kettering. You brought up a very interesting point. Some people present to us as clinician with pain, but on the other hand, we see that some other in the presence of nerve invasion are presenting with numbness or loss of function or paralysis. For example, head and neck, I see facial nerve paralysis with inability to move the face, or you see some numbness. How would you explain this distinct presentation when the pathophysiology is very similar, of basically invading into nerve and interrupting with its normal function?
Yes, I think this may have to do with the type of nerves that the cancer cells are confronted with in each organ. I can well imagine that the type of nerves that are present, let's say in the head and neck area, that are exposed to the cancer cells, probably fulfill some different functions when compared to the visceral autonomous nerves. And the pancreas actually, the sensory nerves do not have, let's say, a direct somatic sensation. They have some autonomic sensation functions. And for, let's say, a paralysis to occur, I think there is no way to detect something like this, let's say a loss of function in the pancreas based on the extent of nerve invasion. Well, at least we can say that when a patient, for example, has pancreatic cancer, in a quite, let's say, large portion, that means the 50 to 60% of the patients, we know that in the first, or let's say in the last 3 years prior to diagnosis, these patients frequently develop diabetes. And it is yet not known how this diabetes occurs in the context of pancreatic cancer. So one question that I always ask myself is that, is this diabetes the result of a dysfunction of the islets due to damage to the nerves that supply these islets? And maybe the correlate of paralysis that you see due to, let's say, loss of motor function in the head and neck area, so that the correlate for that is maybe the loss of the innervation of the islets in the pancreas and the resulting diabetes associated with pancreatic cancer. This is something I can imagine, so sort of the counterpart of such a loss of function. And what is probably still similar between, let's say, head and neck cancers that infiltrate nerves and pancreatic cancer is the emergence of pain. I can well imagine that, for example, also your patients develop pain when they have more perineural invasion. And this is probably due to the fact that the same type of, let's say, sensory fibers get exposed to these cancer cells. This can be, let's say, one explanation, but at least from pancreatic cancer, we know that the pain is more severe when not only in the pancreas, but right behind the pancreas, the nerves are infiltrated, the retroperitoneal nerves. And of course, they have a much higher density of sensory fibers. And I can imagine that when a certain amount of sensory fibers are infiltrated, then the pain emerges. Because we know that the ones with pain are at the same time the ones who have a worse prognosis. So in other words, there's probably a correlation to the amount of nerves that is infiltrated by cancer cells.
Yeah, and you know, you mentioned a very interesting point, the different types of nerves. We're currently so blinded about it. You know, we treat nerves as nerves, which is basically, if I make an analogy for the world of immunology, is like treating all the immune cells as leukocytes or white blood cells, right? It just, from a scientific standpoint, it just doesn't make sense. So I think, what do you think are the barriers for us, the main barriers of cancer neuroscience to make the significant progress into, you know, better understanding of the pathophysiology or neuronal mechanisms underlying cancer progression, so we can actually take them to clinic and target them clinically in clinical trials?
Yes, I mean, that is the holy grail, you know, what you're addressing, you know, that's for the most critical question. I will talk about, or let's say answer this with regard to peripheral cancer neuroscience. So especially for pancreatic cancer, one of the most, let's say, important gaps in my field was the lack of a good in vivo model that shows human-like nerve invasion. And this is something that I focused on in the past 8 to 9 years. And we screened, therefore, many mouse models, genetically induced mouse models of pancreatic cancer that exhibit the same phenotype of a severe neural invasion like we see it in humans. And so we have recently submitted and now got accepted a paper where we described for the first time a mouse model of pancreatic cancer which shows the exact same phenotype of nerve invasion like in human pancreatic cancer. And I feel that such models are increasingly necessary for better understanding the situation in the humans, because in the peripheral cancer neuroscience, although we have good access to, let's say, tumor tissue from a surgical standpoint, we still do not have access to the nervous system of these patients, especially when it comes to ganglia that supply the pancreas. We really do not have access to dorsal root ganglia, which have, let's say, considerable amount of sensory nerves, or let's say sensory innervation. Of course, we have some access to other ganglia, like, let's say, sympathetic ganglia or celiac ganglia, which also provide us with relevant information in this regard. But I think the combination of human-like models that still need to be developed in greater numbers, in my view, and enhanced access and enhanced analysis of human, let's say, nervous system components, these two together will probably enable the actual translational progress in the field.
I wanted to ask you, it's probably a little more broad, right? So I want to ask that, what really inspired you to just, focus on nerve tumor interactions and pain pathogenesis in pancreatic diseases?
Yeah, I think originally, as I said, the inspiration was derived from what I had seen from the research results of my mentor. But today, what really keeps me motivating is the clinical problem of patients who are resected really technically perfectly with a pancreatic cancer, but then 3 months afterwards are presented to us again with a recurrence of their tumor, either locally or systemically, for example, with metastasis. And we know that if a patient has more nerve invasion in his or her primary tumor, this patient is at the same time more likely to get metastasis later on. So the presence of nerve invasion is not only, let's say, a physical reason for the occurrence of tumor recurrence, but it is also a biological indicator for a high probability of tumor recurrence. So I think that beyond this physical aspect of our inability to clear all those nerves in the abdomen that might contain those cancer cells, I think that this nerve invasion suggests to us, or let's say indicates that a cancer that is this well enabled to invade nerves is at the same time a cancer that will very likely spread hematogenously, systemically. And I want to understand the biological reason for the simultaneous ability of cancer cells to invade nerves and also to invade other organs. So what are the mechanisms, what are the machineries in these cancer cells that makes them so fit to infiltrate both nerves and other organs like liver or lung? This is, I think, what really motivates me for future research.
That's awesome. And I want to ask another question. I just, just for me, so are there like a higher propensity of cancers? Like what type of specific cancers have a higher propensity to target the peripheral nervous system?
Yes, that's a very good question. I mean, one class of these cancers has been extensively studied by Moran, for example, head and neck cancers. Actually, the first cancers that were described to have nerve invasion were head and neck cancers. And then later study shows that other cancers, especially abdominal cancers, also exhibit some, or let's say, the different degrees of nerve invasion. And in the abdomen, we analyze many digestive tract cancers, and by far the most neuroaffine cancer is pancreatic cancer. So this is something that we have repeatedly shown and seen, but also other cancers like bile duct cancer, for example, rectal cancer or prostate cancer are also cancers that have a high affinity to nerves. And honestly, we still do not know why it is like that. So we just know that there's a, let's say, grading of cancers with regard to their propensity to nerves, but the mechanisms are still not known.
There you go. No, thank you. It's absolutely fascinating, Ekin, about how cancer cells migrate and interact, can, I guess, put on these different cellular disguises to metastasize and to target multiple types of organ systems, but not just them. They seem to have certain affinity for particular, organ systems. Like lung, likes to go to the CRC. Maybe breast might like to go transmit and travel to the brain. And that has absolutely fascinated me the past number of years.
Yes.
Just saying how cells within the microenvironment find those commonalities and find those sources to be able to survive and thrive.
Exactly, absolutely. Yes, and I mean, as you say, also for hematogenous spread, there are, let's say, preferred organs for metastasis for different types of cancers. That's for sure. And when I talk about pancreatic cancer, I mean, it's of course such that the majority of patients, that means even when we resect them completely, will develop liver metastasis first. But the ones who develop liver metastasis nearly always at the same time also have local recurrence. So these are two phenomena that in the majority of patients occur simultaneously. And so this suggests to us that there's a propensity for this cancer to recur exactly in these two sites, even if you take them out completely during an operation. And and as you say, the same, to the same extent as it's interesting, for example, or let's say exciting to discover why it is like that in breast cancer that it goes to the bones, for example, or why it is like that for let's say lung cancer to metastasize in the adrenal gland. What are the reasons for this propensity? We are looking for similar reasons of propensity now in pancreatic cancer.
Yeah, that absolutely sounds amazing to see how your work is trying to impact and nibble away at that, particularly within the Schwann cells, and manipulating the peripheral glial biology.
Yeah. I mean, Schwann cells, I thought about working with Schwann cell, I would say, maybe 15 or 16 years ago, when I thought that whenever we see nerve invasion, actually cancer cells will probably get into contact not first with the axons, but rather with the Schwann cells that encircle those axons. So this is why I thought that if there is an affinity, probably the cancer cells are more attracted, or let's say primarily attracted to Schwann cells and not to axons, not to neurons. So this is why I said this is worth investigating, and this is why we started with that back then. And the surprise to us was that it was the other way around, that the Schwann cells were more attractive to cancer cells. So they actually migrated toward even the pre-invasive lesions of cancer before really invasive overt cancer occurred. And this notion of a nerve, or let's say neural or neuroglial cells migrating to a transformed tissue, to a, let's say, pre-invasive cancerous tissue, was, I think, also one of the, let's say, foremost contributions that we have made to the field so far.
Yeah, that's amazing. The more you read and the more you become familiar with the field of cancer neuroscience, you find, how much more beyond cancer and more beyond nerves there is there. You started just talking about Schwann cells right now, but very much like what we're seeing in the CNS, variety or myriad of different types of cells that not only provide neuronal signals, but also support the nervous systems are involved in cancer progression. So as we're getting closer to the end of this podcast today, I wanted to ask you, what do you find most exciting about the Cancer Neuroscience 2024 here in Houston, and what you're going to bring and present in this upcoming meeting?
Yes. I mean, so first of all, what I find probably most interesting is that I think it will become, or it already is now, the Cancer Neuroscience meeting that is in the field. So there have been some smaller Cancer Neuroscience meetings in the past, but you, Moran, and your team have managed to bring together all those people who work in cancer neuroscience into one forum. And that's why you also organize this Cancer Neuroscience Symposium, which was a great success with great talks. And I'm sure the second one will be an even greater success, because I know that the field is expanding. There are more people, there are extremely talented people who enter the field, and especially people who are established in other areas of cancer research are also entering the field and providing great data, combining with their major experience. So highly experienced cancer researchers who are originally not cancer neuroscientists are also now in the field. And I think you made a great contribution toward bringing together these people. So it is the forum for, I think, experiencing cancer neuroscience today. And what we will bring in, well, I think there are several topics and several issues that we will bring in, but if I may, let's say, mention one of them, is that a couple of years ago in the central nervous system, it's been shown that the glioma cells, the cancer cells, and the neurons build a functional network, and that the functional network operates over glutamate signaling. And we recently discovered that a similar type of network, again operating through the glutamate signaling, is present in pancreatic cancer. And this is, I think, an important discovery which shows the beautiful actual similarity of some cancer mechanisms present both in the central and the peripheral nervous system. And I think this also gives us the reason to believe that there are really some molecular targets which will be increasingly important for translational purposes. But as I said, this is only one of the several examples and several topics we want to contribute to the symposium. And the rest, with your permission, I would like to keep, let's say, secret until the symposium in February, March. And I hope that we will excite further cancer neuroscientists, further young colleagues for the field with our new findings.
Yeah, that's awesome, man. I want to join your lab now, Ekin. Hearing that you're studying, those things. Thank you so much.
Thank you, Dr. Demir. Yeah, we're very excited actually to have you in Houston, because there's just a really awesome beauty in just, being in a room with your peers and everyone can just talk and learn from each other. And sometimes it's harder to do that over Zoom,
Absolutely.
So I just want to say, thank you everyone for tuning into our podcast. It's been a pleasure to listen to and learn from Dr. Demir. For more information on the Cancer Neuroscience Symposium, please tune in on the next episode. Bye, everyone. Thank you.
Thank you so much. It was a great pleasure. Thank you to you guys.
This podcast is part of a special series highlighting work to be discussed at the 2024 Cancer Neuroscience Symposium, which is hosted by MD Anderson Cancer Center and Wiley on February 28th through March 1st, 2024, with options to attend virtually or in person. Learn more and register at mdanderson.org/Cancer Neurosymposium.